good.
Good. Okay, we're good. I think we're going to get started, folks. Good afternoon, and I'd like to welcome you to the 2015 Cool Climate Oenology and Viticulture Institute's Lecture Series. My name, for those of you who may not know me, is Barb Tatarnik, and I'm the Manager of Outreach and Continuing Education for CCOVI. It is so great to see this many winemakers in attendance today with the emergence of the sparkling wine industry and its popularity. It's so great to see the wonderful support in the room. Today, we are pleased to present Belinda Kemp, who is CCOVI's Senior Scientist in Oenology here at Brock University. At CCOVI, Belinda's work is split between addressing the research and outreach needs of the grape and wine community. Belinda was recruited to join the CCOVI team from Plumpton College in the United Kingdom to apply her extensive research and outreach experience to the Canadian grape and wine industry. Her current projects examine the effects on sparkling wine quality and look at how tannin composition impacts final wine quality. Today, she'll be speaking about press fractions and timing of Bentonite addition on Ontario sparkling wine quality. And with that, I'd like you all to join me in welcoming Belinda Kemp.
Thank you. I just want to emphasize what Barb was saying. It really is really fantastic to see so many winemakers here. It is, after all, studies that are done for you guys. So here we go. So today I'm going to be covering a press fractioning trial we've been doing, continue to do, with Trius at Hillybrand Winery. We're going to look at some preliminary results from that. I'm also going to be looking at the preliminary study of a much bigger study that goes on for several years, but we've got some first results from a protein and bentonite and yeast trial, so we're going to have a look at that as well. And then we're also going to talk as well briefly about a gushing trial that we've got underway with Torres Winery as well, because obviously it seemed a shame not to include that, considering it's sparkling wine as well. So first of all, I just want to say really briefly that all these studies are specifically designed, as Barb was saying, for our wine industry here in Ontario. They're regional specific. They might not necessarily apply to another wine region. And that can be partly because of our clones, our climate. We've got a very short growing season, as we all know, especially this year. And also winemaking techniques as well. So it's applied scientific research. All these projects have come from Fizz Club. Fizz Club is where we meet, the sparkling wine producers meet every 6 months or so, and we discuss issues with regards to sparkling wine, both production and also the latest research that's happening around the world. So I just wanted to reiterate that. So this, the press fraction trial is a continuation of a dosage trial we did with Trius, as in, it's not exactly the same thing, but it sits quite nicely within that field. That was that was funded by NSERC, by an NSERC Engage grant, so I just need to mention that first. As you know, we took the Trius Chardonnay and Pinot Noir blend, and we made a range of different dosages. That's now almost complete. Almost complete. There's a little bit. Oh no, it is complete. I'm sorry, it is completely and utterly finished. We're fine. But that's for another day and won't be included in here. We've already presented the results of that at Fizz Club, as you know. Let's have a look at our press fractions. Now, it's quite common. We know in Champagne that they already do press fractions, and they have their own regulations and rules with regards to that. But just a reminder that if we don't separate the press fraction, and I have seen it, then we end up with low-quality sparkling wine. We do. Everybody knows that. If we separate it out, not only does it give us more ability and more blending options and aging options, but you could also do things like use a second pressing, which is known as the first tie, For things like dosage, again, blending, you may want to keep it as a reserve wine. You may want to use it as a second label. It depends on the winemaker and the quality of that fraction. And then you've got the third tie down here, which can, as I said, can also be used for similar things. But usually that's really quite high in phenolics and the quality is not so good. So again, that depends. What you do, or what the winemakers do with these fractions, depends on the quality and composition of those fractions and aspects like that. So that's briefly what we're doing. And we have taken a commercial grape press, and usually with press fraction trials that have been done, they've been taken at about 12 or 13 stages of the press cycle, of all of the whole pressing, so along the whole set of where the bag deflates and increases, etc. And the point of this trial was it's supposed to represent commercial winemaking. So we've got 3 fractions. We've taken the cuvee, the first tie, and the second tie, which is in practice what would more than likely happen within the winery. Okay. So I've mentioned that about champagne. It's important to mention here that with regards to press fractioning, there's a number of things that will to be taken into account because it can change from year to year, which makes it all the more fun. One can be the volume and composition, and that will obviously depend on things like the pressure of each press cycle, which in turn can be down to the type of press you're using, the length of each press cycle. Some are different to others. Some go up to, for instance, pressure on the first pressing of 1.2, others to 1. The grape variety, because that composition of the grape variety can affect pressing. The wine style that you yourself are after, and the vintage differences as well. So just a few points to remember. So this, I hope you can see this, but maybe it's better on that screen. You can see that in the first glass there, is the cuvee, and the second glass is the first tie. And this is taken from the Trias press. And there's, you'll have to forgive my photography, I'm afraid. Those are our press fractions we started off with. That's to show you. You can see that at the end on the second tie, it's much, much darker. So let's have a look at the press we used. I've highlighted in pink how they've changed the press slightly. So instead of deflating the bag so that we don't let out lots and lots of phenolics and we keep it up, it's only deflated or gone down to 0.2 rather than to 0 completely. And this can be altered accordingly as well on some presses, not all. And we're focusing particularly on this Julce press that we used, or that Trias used. Also, what's really interesting about this is we've only worked on one clone on this, part of this whole study, which is much bigger with regards to clones, bentonite and proteins, and looking at individual varieties and individual clones. So this whole study is on clone 115, which is a Dijon clone, to have a look at that. So we whole bunch pressed. We took samples from each fraction from the tap as it was flowing before it actually went into the tray. So we didn't take it out of the tray at all. So we didn't, didn't contaminate it with any of the other press fractions. We didn't add any enzymes because at the press, we wanted to see the effect of the pressing. We did add 30 ppm of SO2 when we got back here. And the winemaking was all done in triplicate. Because of the time scale of this project, we didn't do any MLF at all. And we did quite a lot of chemical analysis, which is still continuing at the moment. Thank you to Lisa Dowling over there, who's been very busy working on that. We used EC118 yeast for all the fermentations. And we terroired it all and bottled it all at Fielding Estate Winery. We were aiming for 24 grams per litre of sugar. So they're all sitting nicely at Fielding Estates, ready to be disgorged at the moment. So that's the other interesting thing with this study, is the studies that have been done so far don't take it right to the finished bottle of wine. They're always looking at the base wine and don't take it to the finished wine. And that's another aspect to remember about this study. So we can actually taste the difference and see the difference in the final wine. So just some preliminary results we wanted to show you, because as you can imagine, we're halfway through the trial, so there's a lot more to come. So we started off at 18 bricks. The first press fraction was slightly higher. The TA decreased and the pH increased as you went along in the press cycle. That everybody knows that. That's not new news. The nitrogen available to the yeast increased as we went along with the press fractions. Glucose and fructose, a bit boring there. The malic acid in the first press fraction was slightly higher, but depends how important you feel 3.9 and 3.4 grams per litre is. The turbidity level was quite interesting because the second fraction, which is the first tie, was slightly higher. And then the acetic acid, like we all analyse here in Ontario, was of no significance at all, luckily enough. So that's some basic juice of the three fractions going into first ferment. And then after fermentation, the alcohol sits at 10.6. The final one was 10.7. Not sure why, but it is. The TA, or the titratable acidity, decreased, which we expected, and the pH increased, fine. And the yeast assimilable nitrogen on the press fraction 3 was the highest in there, which is interesting when you consider we've got to go into second fermentation as well and make amends for that. Down here on the residual sugar is higher in the third press fraction, mainly due to the higher level of fructose that's left in there, whereas the other two press fractions used up more of the fructose than the third press fraction did. And malic acid, 3, we dropped a little on the malic acid, and turbidity, we increased. And that's really interesting, actually, because usually when you're press fractioning, previous studies have shown that turbidity decreased during the pressing. And that might be because we didn't use enzymes. I suspect it might be because we didn't use enzymes in the before settling. So, but we've got to look into that a little bit further. And a little tiny difference between the final free SO2. We were aiming for between 15 and 20 parts per million when we went to bottling. We were a tiny weeny bit over here, but they're all doing fine and they're very happy fermenting. So we've taken, just to explain the amount of data, we've taken information like this at every single stage before and after we did anything in the winemaking process. So you can imagine we can't fit everything onto here. And as I said, we're still further analysis, chemical analysis still going on. We'll be using the final wines for a tasting of each fraction at the Fizz Club in December. So all our Ontario winemakers can taste the difference between these wines at each fraction. Okay, I'm sure that might not be news to everybody, but it's specific to our region, and that was what was requested. Okay, so our fermentation log of all of our replicate wines there, a bit boring as well. There we go. Nothing exciting happening at all, really. Little tiny bit here of change, but everything catches up. This was monitored by Brix, and then as we got down to lower figures, we used specific gravity. So what's so important about these results? What's relevant to Ontario? Well, one thing, again, I keep stressing, we're not surprised by the TA and pH, but we've still got to have a look at phenolic concentration. We didn't treat any of these wines. We were told that with 115 Dijon clone, that in Ontario, by the time you finish with the in bottle in second ferment, that the phenolics drop out. So we're going to see what happens with the third press fraction and see if that is the case. So we didn't treat anything for phenolics, so we're looking at phenolics, and we're looking at the effect of those on flavour and foaming, because it's been found that higher phenolic concentrations will affect foaming and quite possibly gushing, which I'm going to talk about a bit later. Had a slightly higher level of residual sugar in the second type due to higher fructose levels. How will that affect second fermentation? Because we haven't done anything about it. We haven't changed that. We'll wait and see. I doubt it will have a huge impact. The turbidity increase and change in the second tie, we need to look into a bit more in that to find out what's going on there, because that is quite unusual to have the results that we did compared to previous studies. So foaming, flavour and sparkling wine quality are the further things that we're going to be looking at. Yes, and further statistical analysis, we're going to be analysing the foam before and after disgorging, certainly after dosage as well, and of course tasting it all with all the winemakers. So at this point I would normally ask any questions, but I'm going to save it to the end. So now is another project which is part of a very large project, which is the Omafra University of Guelph Partnership, and this is the first year of the first preliminary, the preliminary study. And this is really regional specific work because it's being carried out on Pinot Noir Mariafeld. Now, for those who aren't aware of this, this is it's a group of clones of Pinot Noir. Seems to be quite a lot planted here in Ontario. It was originally planted in Germany from the beginning of the 90s. It was quite popular. It's quite vigorous, but it's got, here we go here, you can see, nice long loose bunches. It's highly disease resistant. It's got nice big berries, and it's really high acidity, which you'll see in a sec. So we took one container, 200 litres of this Mariafeld, and it was taken from Trius at Hillybrand as well. We had one treatment at the beginning of the juice that had no bentonite treatment at all, and the second one we added 1 gram per litre of Vitiben bentonite to the juice. Vitiben is a sodium bentonite, but we did remove it all before first fermentation. And the aim of this preliminary study, or one of the aims amongst many, is looking at the contribution of grape proteins to foaming. Because I know people are sitting there going, well, why did you add so much, 1 gram per litre of bentonite to the juice going into foaming? Why would you add that? Because in a winery that's really high. And that's the reason we're doing it. We want to really see how important these grape proteins are to the final foamability of the wine. So this is our design. There's our we split the 200 litres into 2 treatments. And then basically when we came to second ferment, that's when the fun began. And we split them up and we used EC118 and no bentonite and our own Brock yeast that has been isolated here, which has been used in previous trials. Here we use bentonite in the second fermentation only, and EC118, and then here we use bentonite for the second fermentation only and Brock isolate yeast. And then we did the similar thing with this treatment here with bentonite as well. So we've got a range, we end up with 8 treatments. To explaining a bit better, the best way is to think of it as a triangular winemaking method. So our first stage got bigger and changed into 2 treatments. We then subdivided it into 4 fermentation reps for the first fermentation, and that was to make sure that we got the same results each time, and we needed replicated fermentations. Then we blended them together because we needed to cold stabilize, filter, and make sure that all the wines that were going into bottle were treated the same at this stage. And then we subdivided it into 8 bottles. So our replicated treatments are all the bottles. We average about 30 bottles per treatment, so quite good for analysis. So we whole bunch pressed in the butcher press over Trius. We did add pectic enzymes to these wines. We added some nutrients to the first fermentation as well. I should have said that about the press fractions. Again, we didn't do malolactic fermentation because of time constraints and things. It was cold stabilized, sheet and plate filtered to 0.45 microns, and then bottled at Fielding, and it's currently sitting there having gone second fermentation. And then we'll be transferring it over to our friend Levi at Maïsme to disgorge, and that will be fun when we've got 8 lots of different dosages to add. And that's another question actually to think about. Some researchers disgorge the wine and just fill it up with the same wine and don't add sugar so that the sugar doesn't cover over the effects of the treatment. But this is a commercial wine trial, so for such a young wine, it's highly unlikely that you would sell that wine or consumers would consume that wine with no sugar in, especially with the acid levels that this variety has given us. So we will do a dosage trial to ascertain which sugar level is best, and we'll use the same sugar level for all of the treatments. I think that's the best way to think. Okay, so our juice analysis on the bento, check out the titratable acidity, whackingly high, 14 and 14, and then it dropped a little here after the bentonite treatment in 1, but that was because we had it at 10 degrees, we were storing it at 10 degrees, so I suspect that's why we lost a little bit there. The pH at 3.1 doesn't matter. The Brix was 19 when we went into ferment. The total nitrogen there, not a huge difference, tiny weeny bit of difference. For some reason, I thought it would lose some nitrogen here, but we ended up with 304 milligrams per litre in the bentonite treated and 284 with no bentonite. The amino acid nitrogen here in the wine was 216 milligrams per litre and 211, and I'll explain why that's important in a sec. Well, I thought it was important. So these are our fermentation logs. Nice little things happening there, nice lines, no difference at all between treatments. I wondered if at 1 gram per litre of bentonite we might have a bit of trouble going in these ferments here, but nothing, no problem at all, no sulphurs, no stuck ferments, nothing at all. That's down to Lisa's care and attention. Oh, and mine. And Esther's. Okay, so I think you can see it probably better on that screen over there. These are the pictures of the wines that had before they, there we go, we can see, you see it doesn't show on there here. So here are the bentonite treatments, and these are all the 4 wines. That's the one treated with bentonite, and this is before first fermentation. And these are the wines not treated. So there's a slight difference, I think you can see it better on that screen over there. There's a slight difference in cloudiness. So the wines on the left are cloudier. It's a bit hard to see on that screen, but it is on the screen over there. So what we're really interested in is how important are our grape proteins to our foaming and our bubbles, because we want those, otherwise we've got still wine. So first of all, we looked at, using the Bradford assay, Esther looked at the initial concentration of grape proteins here, right in the beginning with the juice, before we did anything with it. Then when we added the 1 gram per litre and removed it, you can see a drop here in proteins down here. Sorry. And you've got almost half the amount of proteins in the juice going into fermentation. But after fermentation, they all sort of recover here. There's a little bit here, a bit lower down this end here for this treatment, which is a bentonite at first and second ferment, so that's got a little bit less. But that's probably because of the yeast proteins, the yeast mannoproteins release, that it doesn't show that much of a huge difference, as I said, just down here and here. Okay, so it'd be interesting to see how that correlates with the final foaming in the end. And the final, I should just show you actually, the final one here is the bottled wine. So these purple ones are the wines that that had been bottled straight away and analysed straight away and are sitting in storage. So just before second fermentation kicked off. We use a minimum of 3 bottles for analysis each time to allow for bottle variation from now on as well. So let's have a look, a little bit more information here. So we lost some volume from adding the bentonite. So once before we subdivided into all their treatments, we ended up with 134 litres and 144 litres here for the control. pH 3, TA 11, ethanol nicely 11%, all similar. The only thing that was different with the bentonite added juice was 2 parts per million difference. I don't think that's particularly significant. But when we get down to the treatments that we split up, again, our pH and TA, no differences. This is right before we bottled, okay? Our sugar, we were aiming for 24 grams per litre. We slightly overshot that. Not sure why that was, because we were so careful. But we'll see, because not all of that's going to be fermentable sugar anyway. Our amino nitrogen was slightly lower in the bento, and that's not unusual. If you overfine with bento, you do remove that. You can remove amino nitrogen. But the yeast find that really difficult to use up anyway. They leave it to the end. When they're stressed, they have to take it up, but they have to work really hard to break the nitrogen off to use that. So we're not sure at the moment what that will mean to the finished wine, if anything at all, through second ferment. And our malic acid, check this out, 6, this is the only one for some reason that sits at 6, the others are 7. Quite a high malic acid in this grape variety, in the Mariafeld, which is much higher than clone 115. So possibly prior to going into second fermentation, if it was warmer, if we had more time, we should have considered a malolactic for this one, and certainly we will do next year if we're using this variety, this clone next year. So we're monitoring the wines. We'll be disgorging and dosage, which I mentioned. We'll leave it for 2 months on cork before we start doing sensory analysis here. We've got Esther's working very hard on looking at the protein analysis, not just the concentration, but more importantly, identifying those proteins, basically monitoring those grape proteins right from the beginning, right the way through, so we can see how important they really are, if anything at all. We did do a preliminary tasting at Fizz Club in December of the base wines, and everybody was in agreement. We had 2 completely and utterly different base wines on the flavour front. So it'll be interesting to see what happens. Further chemical analysis, foaming analysis, and its correlation to the protein all the way through, and sensory analysis. All right, so that's that so far. So this Bentonite and sparkling wine is a question that is really topical, not just here in Ontario, but all over the place. And certainly there's been many, many studies done on different varieties, particularly with sparkling wine in mind. We do know that sodium affects Chardonnay and Sauvignon Blanc more than it does Pinot Noir, but calcium bentonite affects Pinot Noir far more than Chardonnay does. Calcium gives far more compact lees, so maybe it's a mixture of both that we should be using for some of our varieties. So that's leading into what I'm going to say about our study that's going to be done this year. So we are with Trius and with Chateau de Charme. We've got 4 varieties. We will have, won't we? They're all so cold hardy, they'll be fine. We'll have a variety of clones of those 4 varieties on 2 soils, and we've got 2 sites for those. We've got sandy and clay soil, so we've got 2 vineyards there. We're going to use sodium, calcium, and a mixture of both, and no bentonite. And we're going to be adding it in the base wine, so fining it, and the tirage, and a mixture of all of those. So that's going to be an interesting project for everybody. So just to explain it a bit better, we'll take our base wine, our variety, our soil type here. We'll split it first into no bentonite, sodium, calcium, and a mixture of both. Then each of those will go off and use different type of bentonite at tirage. So if we took one variety, one clone from one soil, we'd have 16 treatments there. Yeah, and so we're just working out which varieties, which clones. We've still got to discuss that this year, and it is over a few years, so we don't all have to do all of it all in one go. But that's the basis. This is the basis of it. So these circles are the base wine treatments, and these boxes are the tirage treatments. Okay, so we can have a look and see what's best for our region. Moving on to gushing now. So that's our plan for next year. Now, we talked about gushing quite a lot at Fizz Club as well. And there are, if we take all the reasons for gushing from wine makers as well as from scientific papers, we can roughly break it into 3 categories, or I can roughly break it into 3 categories. One is light, UV, particularly sunlight. In this category here, by the way, is bottle handling and disgorging environment. So everything in the winery here and what happens to the bottle. So ambient temperature of the disgorging area, seasonal timing of disgorging. I had one maker saying about never disgorge in winter. Rough handling before disgorging. The angle of the bottle, many machines are slanted anyway, but it depends if you're doing it manually. If you neck freeze the sparkling wine bottles too fast, And the rapid movement from a cold room to a warm room can happen if you don't have temperature control in a winery. So that's that category. If we look at the wine composition, you can include grape variety, vintage variation. People always say vintage variation. We don't actually know how that causes gushing, but we're guessing it's more to do with the composition, chemical composition of the grapes, and whether that's proteins or tartrate crystals that year or whatever. So we're going to look into that in more detail. So protein instability has been implicated, which is strange when you think that you need the proteins for the foaming. The temperature of the wine and the dosage temperature versus the ambient temperature. I should have put down pressure, bottle pressure in there, which can vary between bottles as well. Tartrate crystals, inconsistent mixing during tirage, undissolved sugar in the dosage, although often the gushing happens before the dosage goes in, but it can continue. Yeast from inadequate riddling or disgorging, a high phenolic content. So we know rosés and red sparkling wines can gush more. And another implication is, something else been implicated, is malolactic or partial malolactic fermentation in bottle has been implicated in champagne. And turbidity, which is interesting as well for us because we've got some of that. And finally, packaging materials, cork dust, glass imperfections, and dust in the bottle. So Tor's Winery, we've been working with Tor's Winery, Ben Wiles has, and we came up with a plan, and we were trying to make wines gush. It's not easy because they always gush when you don't want them to. So we tried to mimic riddling cages under UV. Now, I think we did a pretty good job myself. We can't see much of a difference, can you, really? So we cut out the side of cardboard boxes and upended them and left them under UV light in another room at the same temperature as we were disgorging at, in the hope that that UV light would hit them as opposed to natural light. And there's Ben busy loading up the neck freezer there. So that's what happened. And we did it at 4 different temperatures. We disgorged at 10, 12, 14 and 16. And we had wines under UV for all the temperatures and wines under LED lighting for all the temperatures as well. Luckily, some gushed, otherwise the whole thing would be fairly pointless. So what we ended up with is, these are, because we also obviously check the bottle temperatures, which changed during the course of the morning. So these are the bottle temperatures, and this is the temperature down here. This is the number of bottles that were lost percentage, and this is the bottle temperature along here. Okay, so you can see that we lost more at 12 degrees under LED lights, which was quite interesting, than we did along here when we used 10 degrees. So the colder temperature didn't seem to have, oh no, sorry, I'm reading it on the wrong way around. So we lost more, sorry, my fault, seeing it from a funny angle. We lost more at 10 degrees here. We lost far more at 16 degrees under LED light. The UV light at 10 degrees and at 12 degrees had an effect here. But when we got down here to colder temperatures, while UV light didn't have as much as effect as the temperature did. It's really hard to separate in something like this light from temperature. What we do know is the UV light at here, at 16, and the UV light at 12 and 10 had an impact, but so did the LED light as well, which is quite interesting. Well, except 12 degrees under LED light is for this particular wine, in this winery, is the best temperature to use for disgorging it, okay? But it begs the question, why didn't all of the wines gush? Was it because of our lovely handmade riddling cage? Was it because some bottles on the outside got more UV than others? Is it simply because of pressure differences within the wines? Is it because some of those bottles, because of bottle variation, have tartrate crystals or higher phenolic levels or yeast left in them? We don't know. So now that we've got bottles to gush at different temperatures, we can then go on and have a look and investigate what the composition is of those wines compared to the wines that didn't gush. So we're looking at obviously bottle pressure, yeast count to see what's left, malic acid levels to see if there's a difference, and there was some sort of, give us an indication of MLF in bottle. Whether it's protein concentration, but by that we're using, well, heat stability really, does both, but whether it's something, protein has affected it in some way. It shouldn't do, but hopefully we can prove that. Tartrate stability to see if we've got any tartrate still in there. As I said, phenolic, and obviously the standard parameters like pH, TA, alcohol, and residual sugar, and free and total sulfur, so that we know where we are with that. So hopefully, with these analyses, we can find out why those bottles gushed and the others didn't. Okay? So just to finish off before I answer questions, I just want to really thank Lisa Dowling, Esther Onguta, and Ben Wiles for all their help with all this, because you can imagine that with so many projects going on, there's a lot of work to do. It's important to also mention all our industry partners, Trius at Hillybrand, Tours Winery, and Chateau de Charmes for the forthcoming projects that we'll be working with, and Fielding Estate, Ritchie, and Maïsame, because we'll be doing the disgorging with them. All our funding, all our projects are possible because of funding. So the Omafra University of Guelph partnership, the Ontario Centre of Excellence for the press fraction, and the NSOC for the dosage. So that was important. So I'll leave you with that. If anybody has any questions for me. Does anybody have any questions? Do you want us to come and do it at your place? Nothing at all.
Do you want to just go through the pressures on the cycle and the press fraction? Just where the cuts were, and I might have missed it. But.
Yeah. No problem. Excuse me.
And was there anything drawn off the initial, like say the initial 50, 60, 70 meters drawn off as well?
Yeah, we didn't use any of the free one juice. I should have said that.
Okay.
No, we looked at it, and last year they did, but the year we did it, we didn't.
Gotcha.
No, we left that out. So they took it down to 0.2. There's a study that went on in Champagne this year that we mentioned in Fizz Club that just took the first 10 litres of the quality wine coming out of the press after the free run. And then they combined the rest of the cuvee with the first part of the tie. So there's other ways to do it. There's other ways to manipulate. And the idea was to have a premium product here using this wine, and then a higher volume of the second label using half of the cuvee and half of the tie. So big extreme in price differences was the idea. Any other questions?
Actually, I wonder if you could just maybe talk a little bit about the impact that clear bottles for sparkling could have on gushing as well.
It's a bone of contention in the wine industry. Yes, it does. People tend to put rosé wines into clear glasses because they think the consumer wants to see the colour. And there obviously is a truth, but it's about, as Tom Stevenson said, he talks about that. He's a sparkling wine and champagne writer in England and a judge, and he talks about it very much, about why we'll just explain, let them get to grips and learn, consumers, that you're protecting the wine by not using clear glasses. You're maintaining the colour, the flavour, everything in so many levels. So I have talked about it at Fizz Club and to winemakers, and we have been chatting about it, but it's not always down to the winemakers, it's also down to companies and that aspect, and the whole marketing side too. So until everybody gets to grips with it, you can give them the information, but that's as far as we can do. But now that I've got this opportunity, so please don't use clear glass, really. Okay. Yeah, Jake.
One thing that might be interesting, we've gone through disgorging in the last 8 months, and we are taking significant loss on gushing. And as you know, we talked about it, and like Phil, would be interesting to see if Phil and Levi, they, like, what does that cost the winery? And,
Yeah.
What is it? I mean, we're losing probably, what percentage do you typically see, gushing losses? Not for me later than that. So, but is that financial?
Yeah, we can do that. I can do that. We can include that in the final thing. That's no problem at all on the economic impact. Once we got to the higher temperatures, what perhaps wasn't clear in that is that there was more loss from the bottle. The gushing was much stronger and fiercer. So the bottles that are lost, you can put back in and make into a reserve wine, or re-ferment, or whatever the case is. But it's still not going to be released. For the accountant's point of view, it's something that's gone off into the winery until later. But you can't get the wine that's actually lost from the bottle. You can't get back. And if you're doing a great long run and you've got high temperatures and you're losing 30 to 50 milligrams per litre at a time, then that's gone. So, but I can certainly include all the economic costs, financial costs of it, definitely. That's not a problem.
And we'll get a report from LC, use 80 bottles for a 500 or 2,500 bottle run.
And that's what I know. And,
Yeah. Good point.
I knew it with my data. And there is gushing, you can see where there's gushing, there's also how much, another question, how much energy level we use for topping up. Depends how much the bottle is filled in the first place.
Yeah, that's true. We did look at that. We did have a mock system. I should have also explained that. And all the bottles were even on the bottling line, be also right. But the pressure and the sugar, the amount of sugar gone in, aiming for the correct pressure, that sort of thing as well. Yeah, absolutely. But I can certainly do that when we put together the report. Yeah. Yeah.
Just actually a question to Jay, like what practical, looking at gushing it from a practical point of view, like sometimes you just take the cap off the gush, or sometimes and then sometimes you have the dosage that starts to gush. Like and then how much was it gushing? Like half the bottle was full.
Yeah, after like look at our counts when we buy how many bottles they use for topping up, and is that within accepted standards, or was it beyond? And we were beyond. We were using way too much wine. We were losing so much on the gushing. It was just the wines were released for 5 years. We had some challenges with brewing as well. So there's a number of factors.
I'm actually going to do a checklist, sort of reasons and remedies next to it. So like winemakers can sit there and go, right, we've done that, okay, and we've done that, that's okay beforehand. So to minimise the risk of it as well. And so that will be really useful, and that will be available to everybody once we've finished off all the chemical analysis and things.
That's a good point, because if you're adding your dosage and you're actually not adding, if you're adding, eventually it's going on as you're adding your dosage.
Yeah.
And you might not add it if you're trying to add to, say, 15 grams per liter, but you're actually only adding 7 or 8.
Absolutely.
You're using a semi-automatic machine there, but I've done it by hand. I think we had gushing in Canada once, and it was like doing it by hand.
Yeah.
Taking the bottle in your hand, trying to put on the corker, already starts to go in, and trying to put the using a hand corker, and then it
we're doing some work over at Malevoix with that, with Shiraz and Elisa. And we did some manual disgorging over there. And once we moved inside and turned the lights out, we were fine. Couldn't see anybody, but we had no gushing.
Jim, did you have a question?
Well, it's not sure.
Any other questions? So Belinda, do you want to tell folks what's up next for sparkling?
Yes, thank you. So you need to put your date in the diary, June the 9th. It's a Tuesday, isn't it?
Yes, it is.
Tuesday, thank you. We have an international guest speaker, Dr. Fiona Kerslake, over from Tasmania, and she's just she's a co-author on a brilliant review, Viticulture and Sparkling Wine, from the American Journal of Oenology and Viticulture. And she'll be given a masterclass on viticultural effects of sparkling wine in cool climates. She's been working on everything from leaf removal to crop load, a whole range of things on Pinot Noir and Chardonnay. So she'll be giving us that, and there'll be some other guest speakers. Jim, over there in the corner, Jim Millworth will be talking on soils and clones. And Debbie Inglis will be presenting, hopefully, and also George van der Meer, or me. I think I'll be saying something as well. So put it in the diary, and further details of that with regards to the itinerary and things like that, and costs, etc., will come out nearer the time. But just to make sure that you put in your diary and hold on to that date and join us for that.
Yes, because that one's open to everyone.
It is. It's open to everybody. It's not just FISCAP members. It's students, grape growers, lecturers, anybody and everyone.
So basically from vine to wine.
Oh, nice. Yes,
There we go.
fine to on.
Okay. So again, thank you very much for coming. Thank you, Belinda. We have a small gift for you. And this isn't a cork or a sparkling cork. It's actually a USB, but thank you so much.
Just to show you. Oh, hang on. Yeah, look.
So again, please join me in thanking Belinda. This was a great talk and a timely subject. Thank you, Belinda. Just so everyone knows, before we head out, our next speaker actually will be Dr. Jim Wilworth in the back of the room, our CCOVI viticulturist, and his topic is optimizing cold hardiness and winter survival in grapevines based on trials and experiences here in Ontario. So we'll get to hear some of the latest news from Jim. So again, thank you everyone for coming out, and we'll see you again next week. Never know which button to push.
You hit that, or end, or if you hit that.